Thank you for giving me this opportunity to talk about my research interest on this platform. And I hope everyone can hear me. Good afternoon, good evening, or good morning, whoever from wherever you are listening this webinar. And thank you for joining us. So my research interest is about post-translational modification. I work in especially in SUMOylation, and I'm going to talk about what we have discovered in last few years in this research area. So before I start my talk, I would like to thank the people involved in this whole research, whole discovery, and especially these previous students of Ari's lab, postdocs, masters or graduate students, but especially these peoples such as Sunjin, who stands for me every time for transforming the rice and is the transformation guy who can transform everything. And Mark and Beatrice, when I joined the Ari's lab, they were already there. So I would like to thank for giving me their hand, their help during my research, as well as then later on Vivek joined as a postdoc, and Momita and Alberto is a current postdoc. They are really helpful, and thanks to them. Of course, Ari, as well as our collaborator, such as Professor Malcolm, Julie from Sheffield, Christoph from CIRAD, and Lars from JIC, Mureg from Warwick, and Michael. And of course, I should not mention about the funding agency, that is, they pay money for my funding doing research, that is ERC and BBSRC. So before what in Ari's lab or we are trying to do, trying to understand is about how we can, the mechanism to provide the tolerance, or you can say make the plants more resilient against the biotic and abiotic stress. And the tool we are using is post-translational modification to understand the mechanism, underlying mechanism for growth and development of plants, or how plants deals with abiotic stresses or biotic stresses. And these post-translational modification, there are various different kind of post-translational modification is known already in plant and animal system. And in short, if I say once protein synthesized, they get modified with the addition of either chemical group or a small polypeptide group, they get modified, and most of the time these processes are reversible. So you can this modification, this modified protein can be unmodified or removed. This modification can be removed with the special enzymes, and this modification we are especially involved in understanding the process of SUMOylation. And we have shown that this SUMOylation or any other post-translational modification can affect the localization of protein, where you can see example of this here. This is the benthamiana. We have done some microscopy to see the protein of a protein which is a wild type protein, and when we mutated the SUMO, where make them unable to conjugate with the SUMO, you can see the pattern of the localization in the nucleus. They are forming, it changed, and they are forming the spicules, nuclear spicules. As well as we have shown in this year, we have shown that the BZR1 transcription factor, their localization from cytoplasms to nucleus is regulated through the SUMO conjugation and deconjugation. As well as we have also shown that protein-protein interaction gets changed once you have SUMO mutate the protein where they are not able to conjugate with SUMO. So the interaction with a specific protein change where the wild type can interact very well. So it's not only in plants, it's in animal system, it's very well known. And recent review from Edward Lab have shown that how SUMO conjugation with the help of different enzyme and deconjugation of with the help of different enzyme can affect severely from in different diseases such as heart diseases, as well as cancer or neurological diseases. So the significance is very well established in the plant system. So we wanted to know how this SUMO is working in and can be utilized in crop improvement program, you can say, and how we can enable the plants to cope with the biotic and abiotic stresses. So before I show you some data, how we did it, I want to show that what is exactly SUMO. So SUMO is a small protein, 10 to 12 kD Dalton protein, and these proteins in Arabidopsis, if we talk about Arabidopsis as a model system, we have identified 8 different SUMOs, but not all the SUMO has been characterized yet. So still a lot of things has to be done in this field, and if you we know about more about SUMO 1, 2, 3, and 5. If you focus on this sequence alignment, you can see that on C terminal they have a diglycine motif, and this diglycine motif is very important when SUMO is going to conjugate with a target protein. So if this SUMO has to conjugate on the protein, it has to be cleaved on their C terminal to release to expose this diglycine motif. So what happens really in the process, SUMO diglycine motif, SUMO synthesized as a premature, you can say premature SUMO, we call it as a premature SUMO, and they gets cleaved off the C terminal, cleaved off by the SUMO proteases. Once SUMO protease cleave off the C terminal, diglycine motif gets exposed, and this enables it to conjugate with the specific lysine, where a specific enzyme, SUMO E1 activating enzyme or conjugating enzyme, as well as E3 ligase, play a role in conjugating this SUMO to to the target protein. But this lysine cannot be any lysine. It's a specific motif, must be in a specific motif, and that classical consensus SUMOylation motif, or you can say site, is very well investigated 20 years back from Ronald lab, Edinburgh, and he has shown that this is the classical motif where you can see lysine and aspartic acid and glutamic acid is required, surrounded by the aspartic acid and glutamic acid. And the challenge in this SUMO field is identifying this lysine is the biggest challenge what we face. And we wrote a whole program, and we have a software to identify this lysine, specific lysine in a substrate. So if you can find, and recent study has shown that this SUMO conjugating motif can be different variant in different variant form. So it's challenging part in the SUMO conjugation and SUMO field to identify the lysine. So once you identify the lysine, you can see how they are working. But in our lab, we are not focusing on SUMO conjugation part, because there are quite a few reasons. One reason I can mention at least here, that the plant E1 and E2 and E3 enzyme is only one enzymes for E1 and E2 known, and whereas E3 only 2 enzymes till now we know in the plant system. So whereas SUMO protease, we have identified a different whole family of the SUMO proteases in Arabidopsis, 2 different families. I'm going to come back later on that. But in rice, they are more diverse, means in crop system, they are more diverse. And we are very much focused in the lab in SUMO protease form, that how this SUMO protease acts or cleave off the SUMO from the target conjugated form of the substrate to free the SUMO. So basically, we are very much focused in this area. And we wrote quite a few review in last few years as we get the information, as we reveal the information, we try to convey in the society in that form of review. And we wrote quite a few review. If you want, you can go to these reviews. So, and as I mentioned, we have identified a different family of SUMO proteases in Arabidopsis, and we can classify them into 2 different families. One is broadly in 2 families, basically. One is centrine type of proteases, or ubiquitin like SUMO proteases. All the SUMO proteases, the notable point is all the SUMO proteases we have identified are all cysteine proteases. So as cysteine proteases have contain the cysteine along with histidine, and in this sentient type of SUMO proteases, aspartic acid is the part of. So we look for this motif, or you can say catalytic domain, to identify the SUMO proteases, ULP type of SUMO proteases. And we have identified 8 different SUMO proteases in this class, whereas other class we have identified it recently in 2018, where DESI type of SUMO proteases. This is desumulating isopeptidase, where cysteine and histidine is conserved, but it along with the asparagine. So we have identified different kind of DESI SUMO proteases does different thing. And all these SUMO proteases, what we have identified till now, we have shown that they do different things involved in different different morphology or different action of the in plant system. So how you are going to see, how we can see the SUMO conjugation and deconjugation basically in our lab. So in the process, you can you see that this SUMO is available and the substrate is available in the cellular system, and enzymes are available. So what we do, if you put the stress on, like here is the example is NaCl, sodium chloride. When you start increasing the stress, you can see that you are pushing this process to conjugate. The SUMO gets conjugated with the specific lysine or specific substrate lysine. So you can, and when you do the Western, extract the total protein and do the Western blot and probe with the SUMO antibody, you can see the SUMO conjugation gets conjugated protein start appearing. But as soon as and SUMO protease is there in wild type to cleave off some, but once you have SUMO protease mutant, you can see even without stress, you can see the equal level of SUMO conjugation in absence of SUMO protease. Means loss of function is all showed that if you don't have SUMO protease, you can see the SUMO conjugation in the sample without any stress. And as soon as you start increasing the concentration of salt or stress, you see more conjugation of SUMO, and that tells, and that's how we detect the SUMO conjugation and deconjugation. In the last few years, we have discovered the role and significance of SUMO proteases in plant system. And Beatrice was the previous postdoc of Ari's lab. She showed that how SUMO deconjugation and conjugation can regulate the lateral root formation depending on the water availability. Whereas Momita is a postdoc in Ari's lab, showed that how SUMO conjugation and deconjugation can regulate the protein or specific protein transport from cytoplasm to nucleus. Later on, we have also shown Vivek, previous postdoc, and now he's running his own lab in India, and he showed that previously it was known that WRKY33 is for WRKY33 phosphorylation is required for their activity. But what Vivek have shown recently, and the paper is in PNS under revision, he showed that SUMOylation of WRKY33 is required before phosphorylation of WRKY33. So if you don't SUMOylate WRKY33, you don't get the phosphorylation of WRKY33 to activate the signaling pathway during to provide the plant immunity. And I have shown in a couple of years back, I have shown that how jasmonic acid signaling can be modulated with the help of SUMO conjugation and deconjugation. Here I have shown that JAS repressor, which negatively regulates the jasmonic acid signaling, if you SUMOylate the JAS, SUMO gets conjugate with the JAS, it inhibits its interaction with the COI1, which is a F-box protein responsible to degrade JAS. It SUMO inhibits in its interaction. So once it's inhibited the interaction, JAS can accumulate, and its accumulation suppress the MYC2 activity, and that's how we can modulate the plant immunity in jasmonic acid signaling dependent manner. Since we have all this information and all this data in the Arabidopsis, we wanted to translate the information and knowledge into the crop system. So we started looking, and rice is an important crop, we all know that, and 50%, more than 50% population of the world depending on the rice. So we started looking how SUMO proteases are evolved in rice system, and we have shown that when you modulate the activity of the SUMO protease, a specific SUMO protease, you get the root growth and development, different types of root growth and development, and you can enhance the root growth development in rice plants as well. And very recently, from JIC with the Lars group, he showed that one of the SUMO protease, heartbreak SUMO protease, if you have mutant, you don't get the fruit development. So this is basically Brassica species, and we can utilize these rice or this fruit development in Brassica species. We can utilize this for crop improvement program. So we have a hope. We can assume that if we study better way, we can utilize this SUMO protease or SUMO modification system in crop improvement program. So before we believe, most of the people believe that all the SUMO activity or SUMO machinery, you can say basically, or component, you can say basically, are available only in the nucleus. Basically, it's all activity is nuclear localized. It was the, you can say, myth that all the activity is localized in the nucleus. But our target, you can say, all the targets are only nuclear localized. But we have we break this, and we discovered that in 2018 that these SUMO machinery can be also available to do the to desimulate or simulate desimulate the proteins which is localized in plasma membrane. And later we have shown that SUMO proteases are available in cytoplasm as well to desimulate the target protein. So, and I'm going to talk about this story today in later in this talk about Brassinosteroid signaling, how we identified this cytoplasm localized SUMO proteases. What we have done, we try to characterize all of the SUMO proteases in the lab. So during the characterization, we identified a SUMO protease which is we found in cytoplasm localized. So once we saw that, we looked into the literature whether someone can someone already identified it or not, and we found that George Copeland lab from Max Planck had already identified this SUMO protease and characterized in different ways. And we started looking at this SUMO protease, and we found that these SUMO proteases are involved in brassinosteroid signaling. When we put them in the brassinosol, which is an inhibitor of brassinosteroid biosynthetic pathway, this SUMO protease ULP1a is less sensitive to this inhibitor. So then the question came in the mind that definitely in brassinosteroid signaling pathway, there is some kind of substrate or protein working as a substrate, which is simulated. And this SUMO protease, which we identify working in brassinosteroid signaling, should be working to desimulate this path, this target. So the question was, basic question was, or you can say obvious question was, what is the substrate and what is the condition they are getting simulated? And of course, if they are simulated, what is the significance of this simulation? To answer this question, we started looking for the into the brassinosteroid signaling, and we know the significance of brassinosteroid signaling by looking at this phenotype, the wild type and BRY1, which is a receptor of this brassinosteroid. If you have loss of function or mutant of BRY1, you can see how different they are. BRY1 is not able to even grow them. So this brassinosteroid signaling, you need them for a proper plant growth. And we looked into the different component of this brassinosteroid signaling, and we know that BRY1 is a receptor, and this receptor through the different cascade of reaction, it suppresses the BIN2. BIN2 is a kinase which phosphorylates the BZR1 transcription factor, and this BZR1 with transcription factor get phosphorylated with the help of BIN2. Until that time, we knew that BZR1 gets phosphorylated and gets ubiquitinated. And BZR1, once it is dephosphorylated form, it can go to the nucleus. So we started looking all the components, as a SUMO target, and we run those proteins in our software. As I mentioned before, that we wrote and basically a PhD student, Stuart Nellis, wrote that program, and we run those all the protein, and we did some bioinformatic analysis, and we found that BZR1 is the potential target for SUMO conjugation. And then what we did, the first thing we did is a transient assay. And in transient assay, in Benthamiana leaves, which is a co-immunoprecipitated with the GFP tagged version, and we have identified the lysine in BZR1. So we mutated that lysine into the arginine, and this and then co-infiltrated BZR1 with SUMO and BZR1 2KR with SUMO. So we found that when you probe with the SUMO, you can only get the SUMO conjugated BZR1 in wild type, but not the BZR1 2KR. And in addition, we have shown that when you treat this, and we generated basically the transgenic, when we treat this BZR1 transgenic plants, we can see that the hyperconjugation of SUMO, hyperconjugated SUMO BZR1 appears when you treat them with the brassinosteroid. Brassinosteroid that activates the brassinosteroid. It's a chemical form of the brassinosteroid. So when you treat brassinosteroid, you get the hyperconjugation. It tells you that when you have a brassinosteroid, active brassinosteroid signaling, it gets SUMO conjugated. So what happens, to answer that, we knew, as I showed you before, that once this is BZR1, once this is modified, what is happening? The question was how they are modulating the action of BZR1. So there was three possibility, basically: localization, stability, and activity. And we started looking at all of them. So we looked into, if you look at this, the phosphorylation, the possibility was either BZR1 is not getting phosphorylated, or if this is not getting phosphorylated, it cannot be, it's not interacting with BIN2. The first possibility was how the interaction is getting modulated between BIN2 and BZR1. So when we looked into it, the wild type BZR1 interact BIN2, it's known, but when we have desumolatable form of BZR1, it interacts more. So basically, you have desumolatable form, you can interact more, and you can phosphorylate and ubiquitinate. But when you have SUMO on it, you cannot interact that much as you can interact without SUMO. So we started looking for the localization of the protein, and we generated the transgenic, and we treated them with brassinosteroid to activate the brassinosteroid signaling. So basically what happened, when you have a brassinosteroid, you can see the accumulation of BZR1 in nucleus gets increased, whereas this is not very well seen, or significantly less in 2KR form. So that shows that SUMOylation is required for the translocation or nuclear accumulation, you can say, from the nuclear accumulation of BZR1. Then later on, we started looking if BZR1 is the right target for the SUMO protease we have identified, the question from where it raised. So we checked for the interaction between BZR1 and ULP1a, and we found that they physically interact. Basically, they interact to desumulate this ULP1a SUMO protease, interact to desumulate the BZR1. So 2KR is doesn't interact at all with the ULP1a, and we have done a in vivo analysis where we have shown through the microscopy that you have they co-localize together. ULP1a and BZR1 co-localize together. You can see some since BZR1 localized in both nucleus and cytoplasm, you can see they are localized in GFP tag, BZR1 localized in both places. And when you overlay them, you can see they are coming together. Whereas when you treat them brassinosteroid, that is activating brassinosteroid signaling pathway, it degrades the ULP1a. So you can't see any ULP1a here. And whereas BZR1 goes into the nucleus. And then when you have BRZ, which is an inhibitor of brassinosteroid signaling, you get accumulated ULP1a and ULP1a. and then all the BZR1, most of the BZR1 comes back, stay back in the stay back in the cytoplasm, and they could co-localize together. So they showed that when the brassinosteroid signaling is active, it degrades the ULP1 to simulate the BZR1 and activate the brassinosteroid signaling. So you can see here in this model, which we propose in the paper, that BZR1 in normal condition, or you can say non-stress condition, BZR1 is in simulatable form, whereas ULP1 suppressed, is not accumulated. Whereas as soon as stress comes, ULP1 gets accumulated, and that cleaves off the SUMO to suppress the growth. So that's what we have shown in this paper, how in salt stress condition and non-stress condition, how SUMOylation is regulating the plant root growth. And so ultimately we answered the question that who is the substrate, and in what condition is getting simulated, basically active brassinosteroid signaling, and that enables the plants to grow better under salt stress. In the end, I would like to show a few slides for our crop improvement program in Durham Crop Center. We do potential how this SUMO Protease potential can be utilized. And we have identified, as I mentioned, we have identified a broad family member in rice compared to the Arabidopsis, and we have shown that different SUMO proteases, we have identified the different SUMO proteases in rice plants. And later on, during the characterization, we have shown that and made some overexpression line for gain of function and loss of function for SUMO protease OTS overlay to tolerant salt. And we found that if you don't have any stress, if you grow them in normal condition, you don't see any difference in the root length. But if you put them under salt, you can see the OTS RNAi line, or you can say loss of function of OTS, makes them susceptible against the salt. And not only that, when you grow them in these RNAi line or overexpression in the normal condition in plant, in the seedling stage, we didn't see any difference in the salt. But in normal condition, when we grow them in the pot in soil, after 45 days, we have seen that difference, that overexpressor is going much deeper than the RNAi line or vector control. And we have generated the CRISPR lines as well, and we found some kind of deletion and insertion for the OTS gene, and it's all has been done from the Sunjin, who does all the transformation and all the transformation for rice and other transformation as well crop, and he runs as a transformation facility in Durham basically. So what he showed that OTS1 CRISPR mutant also have showing the same phenotype what we got with the RNAi line. So you can see here that in the normal condition, when you grow them in MS, you don't see any difference between the wild type and CRISPR mutant, but when you grow them in salt, you can see the shorter hypocotyle, or you can say seedling height, as well as root length is shorter. And we did some drought stress experiment as well, and we found that the OTS1 overexpression having the deeper root length have the drought drought susceptible, and whereas this is drought tolerant, OTS1 RNAi line showed the drought tolerance. So we wanted to see the mechanism, how this mechanism working, why they are susceptible is just opposite to the salt stress response, and we wanted to see if they have some kind of stomata different, stomatal number of stomata is different in different transgenic, and we did not find any difference in and this work has been done in collaboration of Professor Julie Gray and Bobby worked really hard to find out this there is no stomatal density difference. So then we start looking at the literature, and we wanted to see what is the mechanism behind this drought tolerant behavior of SUMO of this SUMO protease mutant. So we found through the Is2 hybrid analysis, we found that a target BGP23, which is known as a responsive gene for the drought tolerance in rice, and we showed that if you do the in vivo analysis, BGP23 gets simulated or SUMO conjugated. So later we wanted to find out whether BGP23 status of BGP23 in overexpressed RNAi line. So we found that, and we showed that in RNAi line, BGP23 gets simulated and accumulated. So basically BGP23 accumulation basically providing the drought tolerance because it's getting simulated and conjugated to SUMO and stabilized in RNAi line, and that makes it more drought tolerant. So we put the model on, propose the model that in drought, the SUMO protease gets degraded, and this BGP23 kind of target, which is responsible or positive regulator of drought response, they get SUMO conjugated, and this SUMO conjugation leads to its accumulation and promote the drought tolerance. So in ultimately, I can conclude my talk. I think I'm running out of time, so I can conclude this talk as like SUMO is required and pivotal for in plant growth and development during stress, and these SUMO machinery can be utilized in crop improvement program. And in last, I would like to announce that I'm moving from here to Nabi, back to India, and positions are open to join the team, and I'm happy to collaborate. So please contact if you can, if you want to work, and this is my Twitter handle. So if my email change, you can just contact me on this as well. And thank you all for listening, and I'm happy to take the question.
Excellent. Thanks very much, Anjil. That's very interesting talk indeed. I think things will look a little different from Durham Cathedral when you move back to India, I'm guessing. A little bit of a different environment there, that's for sure. So let me encourage everyone to please ask any questions if you'd like. We have a couple come in already. So first question from Jitindra Giri, and they say SUMO is everywhere, and it seems to be the case. And then they ask, is there any bias in the roles in plant function in particular? And if we knew that, that would help with targeting a specific trait for improvement. So are there any biases in the pathways that SUMO plays a role in?
I wouldn't say it's a bias, because means if you ask me as a SUMO guy, I would say all the SUMO protein gets simulated, but the challenge is we need to identify what condition and why. So I really doubt there is a biasness, but yes, we need to still look to answer this question. I would say still a lot of things to be done, and I really can't answer yes or no in this situation.
So I mean, in Ari's lab, a lot of the work you do is about salt tolerance or drought tolerance.
Actually, we do a lot of biotrophic infections as well. I didn't show a lot of biotrophic, but basically we do a lot pathological stuff as well. So yeah.
So you're finding SUMO in all sorts of different pathways.
Oh yes. Sure. I mean, so we found we have showed. I can tell you this, that in all the phytohormone signaling, we are in the position to tell you in all phytohormone signaling pathway, there is a target we have identified.
Okay.
So you can imagine, like it's in the salt, in the necrotroph, in biotroph, drought.
Okay.
Yeah.
So I had a question about the CRISPR mutant that you developed, the rice CRISPR mutant. So it looked, when you showed a picture of it when it wasn't in drought conditions, it showed as if it was a little bit smaller. So do you get a change in production in that line?
We never looked into further. Actually, we looked into the RNAi line in Plant Journal paper, the one I showed you in this paper. For RNAi line, there was RNAi line was better productive than the overexpressor line. but we never take this CRISPR paper to that level. No.
Okay.
Yeah.
Okay. I see. Okay. Okay. So I'm going to have another question, I think, if I may, and think about the evolution of SUMO. So you have mentioned, and this obviously was picked up in the other question, that SUMOylation is involved in lots of different processes that we just discussed. So is there any evidence that this is a recent evolution within plant systems or other eukaryotic systems, or is it there in lower plants as well, SUMOylation, or is it more prevalent in higher plants? How does that work?
That's like my future question he asked.
Okay.
There is, and I'm writing the grant as well, so definitely there is.
Okay, so there is SUMOylation in lower plants.
Evolutionary aspects in SUMO as well. Yeah, definitely.